This is the 13th video in the fundamentals of biology, and today we're talking about gene regulation. So in prokaryotic cells, they will have something called operons. And what is operon? It is a group of genes that will transcribe as a group. So in this operon, gene 1, gene 2, and gene 3 will all be transcribed through one promoter. So a single promoter will control these genes at the RNA polymerase binding site. And regulatory DNA sequences are binding sites for regulatory proteins. And repressors will bind to operators and will block RNA polymerase, so transcription cannot occur. So in this case, a repressor is binding to something called an operator, and this promoter is unable to transcribe these genes. On the other hand, activators will bind onto the DNA, and this will help RNA polymerase to bind to a promoter, and this will increase transcription. Here's some more about regulatory proteins. Regular proteins themselves can be activated on or off. Regulatory proteins are made by regulatory genes. And small molecules called activators can turn regulatory proteins on or off. If it's on, it is able to bind to the DNA in order to affect transcription. On the other hand, if it's off, it will not be able to bind to DNA and will not affect transcription. One type of operon is the Lac Operon. So in order to use lactose as energy, bacteria such as E. coli must express the Lac Operon genes, but it will only express lac when lactose is available but glucose is not available. So this entire operon contains a lac repressor and a catabolite activator protein, or CAP, in order to measure glucose. When lactose is not available, the lac repressor will bind to the operator in order to prevent the transcription of genes. However, when lactose is available, something called allolactose, which is basically lactose but rearranged, will bind to the lac repressor, and this will cause it to leave the operator and will allow for transcription to occur. So in this case, allolactose is something called an inducer and will trigger expression. And the Lac operon is inducible because it is usually off or repressed, but is able to be turned on when lactose is available. So to summarize in this picture, when lactose is not available, the repressor will be able to bind to the operator, whereas if lactose was available, the repressor will not bind to the operator. When glucose is not available, CAMP will be produced in order to attach at the CAP. As a result, it will bind to something called the CAP site, which will help RNA polymerase to bind to a promoter. This will help transcription occur when there are low levels of glucose. However, when glucose levels are high, the CAMP is not made, so CAP is not able to bind to the DNA. So therefore, when there is a high level of glucose, lactose will not be made because E. coli will usually depend on glucose first before relying on lactose. Another type of operon is the trip operon, and this is a repressible operon. So usually E. coli can make the protein tryptophan if it's not in the environment with the trip operon. The trip repressor will prevent the transcription of genes. So if tryptophan is present, the tryptophan itself will attach to the repressor in order to activate it, and this repressor will prevent transcription and is called a co-repressor. However, if tryptophan is not present, the trip repressor will be inactive, so the RNA polymerase is able to transcribe these genes at the end. Eukaryotic gene regulation occurs in many places, and this can cause differential gene expression. This is where different genes are expressed within the same genome. So the first way genes are regulated within eukaryotes is through the chromatin structure. Something called histone acylation will add an acyl group to histone tails. And what this does is it opens up chromatin structure in order to promote transcription. The opposite of this is called methylation, which is where you add methyl groups to DNA, and this will condense chromatin in order to reduce transcription. The affecting of chromatin structure is known as epigenetic, because it does not affect the actual DNA sequence. Instead, it will either open up or condense the chromatin. Transcription initiation is another way genes are regulated. So control elements are non-coding DNA and are the building sites for transcription factors. If they are located near the promoter, they are called proximal. However, if they're far away, they are called distal, and they are also known as enhancers. Because they are far away from the promoter, they are known as upstream from the gene. An example of this are activators, which bind to enhancers in order to promote transcription. This process is also helped through mediator proteins, which will position the pre-initiation complex. In RNA processing, there can be an alternative splicing of exons and introns. So depending on how spliceosomes decide to splice the pre-mRNA, they will create different RNA sequences in order to produce a protein. Next is RNA stability, and miRNAs or microRNAs can cause mRNA to be chopped up, decreasing its lifetime. And finally, translation can be regulated, and this is where miRNAs can block the process of translation. Thanks for watching this video. This lesson covered operons, regulatory proteins, and eukaryotic gene regulation.